Deep into the past, millions of years ago, a tale started. A tale that would lead to the creation of the most complex creatures on our planet, us. How did we originate? What secrets do our ancient beginnings hold? Imagine a world where our early ancestors roamed, encountering challenges of life and death. They were not solitary. The Earth was a battlefield of survival, where only the fittest, the cleverest, and the most flexible could survive. Who were these early creatures that walked the same paths we do now? As researchers discover ancient remains and objects, they offer intriguing glimpses into a past that's hard to fathom. Every find, a piece of the puzzle, creates a clear image of our journey of evolution. What do these artifacts reveal about the creatures that lived before us? How did they evolve into the modern humans we see in the mirror every day? The tale of human evolution is full of surprising twists, remarkable changes, and significant findings. From the early hominids with their crude tools to the emergence of Homo sapiens, our forebears faced incredible challenges. What motivated their evolution? What environmental forces shaped their destiny? Get ready to be amazed as we explore the ancient world, discovering secrets that have been hidden for thousands of years. This isn't merely a history lesson, it's an enthralling story of survival, innovation, and change. How did our forebears develop the intelligence, creativity, and resilience that characterize us today? Join us as we peel back the layers of time, revealing the captivating story of human evolution. The clues are spread across the world, waiting to be assembled. What will we learn about our ancient heritage, and what does it mean for our future? The process of human evolution is how Homo sapiens came to be a unique species from the hominid group, which encompasses all great apes. This slow progression included characteristics such as walking on two legs, fine motor skills, and advanced communication, and involved mating with other hominids, showing that the evolution was more complex and interconnected than straightforward. The exploration of where humans came from, known as the study of human origins, is covered by various fields of science, including physical and evolutionary anthropology, paleontology, and genetics. Primates split from other mammals around 85 million years back during the late Cretaceous era, with the earliest primate fossils dating back over 55 million years to the Paleocene. Primates evolved into different groups, leading to the ape superfamily, which then developed into the hominid and gibbon groups. These groups split about 15 to 20 million years ago, with hominids, including orangutans, splitting from each other around 14 million years ago. Hominids, which include Australopithecines and the Pan genus, split from the Gorillini tribe, gorillas, between 8 and 9 million years ago. Australopithecines, which include the extinct two-legged ancestors of humans, split from the Pan genus, chimpanzees and bonobos, between 4 to 7 million years ago. The Homo genus came into being with H. habilis over 2 million years ago, and anatomically modern humans appeared in Africa around 300,000 years ago. Before Homo. Early evolution of primates. The history of primates stretches back 65 million years. Among the earliest known primate-like creatures are Plesiodopus from North America and Archicebus from China. These early primates thrived in the warm, tropical environments of the Paleocene and Eocene periods, covering vast areas of Eurasia and Africa. David R. Begun suggests that these early primates were dominant in Eurasia, with a lineage that eventually led to the evolution of African apes and humans moving south from Europe or Western Asia into Africa. The group of primates that survived in the tropical regions, as seen in the Fayum Depression near Cairo, eventually evolved into all modern primates, including lemurs, lorises, galagos, and anthropoids, which include New World monkeys, Old World monkeys, and great apes, including humans. The first known catarrhine, Camoyopithecus from Kenya, is dated to 24 million years ago, with its ancestors including species like Egyptopithecus and Propleopithecus from around 35 million years ago. In 2010, Sedanius was identified as a relative of the last common ancestor of crowned catarrhines, estimated to be around 29 to 28 million years ago, bridging a gap of 11 million years in the fossil record. Around 22 million years ago in the early Miocene, basic catarrhines from East Africa indicate a long history of diversification. Fossils from this period include Victoriopithecus, the earliest known Old World monkey. The ape lineage, up to 13 million years ago, includes Proconsul, Rangwapithecus, and others, all from East Africa. Fossils such as Otavipithecus from Namibia and Dryopithecus from Europe show a wide variety of forms during the warm climates of the early and middle Miocene. The most recent Miocene hominoid, Oreopithecus, is dated to 9 million years ago from Italy. In 2023, a study on Anadaluvius Turkey from 8.7 million years ago proposed that hominids originated in Europe, with the oldest known hominines being European and migrating to Africa before 7 million years ago. Molecular studies suggest that gibbons split from great apes around 18 to 12 million years ago, and orangutans, subfamily Pongini, diverged from other great apes about 12 million years ago. The subfamily Hominini, African hominids, branched off from Pongini approximately 14 million years ago. Hominins, which include humans and the Australopithecine and Panina subtribes, branched from the Gorillini tribe, gorillas, between 8 and 9 million years ago, with Australopithecines separating from the Pan genus, chimpanzees and bonobos, 4 to 7 million years ago. The Homo genus emerged with H. habilis over 2 million years ago, while anatomically modern humans first appeared in Africa around 300,000 years ago. Divergence of the human clade from other great apes. Genetic evidence suggests that around 8 to 4 million years ago, gorillas diverged from the human lineage, with chimpanzees, genus Pan, following closely. Human DNA is about 98.4% similar to that of chimpanzees when looking at single nucleotide polymorphisms. However, the fossil record for gorillas and chimpanzees is scarce, likely due to the poor preservation of fossils in acidic soils of rainforests and a bias in the way fossils are collected. Other early human relatives likely lived in drier areas beyond the tropics, coming into contact with animals like antelopes, hyenas, dogs, pigs, elephants, and horses. As the tropics began to shrink around 8 million years ago, there's a lack of fossil evidence for the split between the human lineage and that of gorillas and chimpanzees, which is thought to have happened around that time. The earliest fossils that might belong to the human lineage include Sahelanthropus cadensis, 7 million years ago, and Ororantogenensis, 6 million years ago, followed by Ardipithecus, 5.5 to 4.4 million years ago, with species A. r. cadaba and A. r. ramidus. A study on the early life of A. r. ramidus reveals a unique combination of physical and behavioral traits in early hominins that are not seen in any other great ape. The study found that the skull shape of A. r. ramidus is similar to that of young and juvenile chimpanzees, indicating a more juvenile-like skull shape due to differences in growth patterns. The study also suggested that early hominins, similar to bonobos, Pan paniscus, might have evolved through self-domestication, challenging the traditional chimpanzee referential model. This suggests that it's no longer valid to use chimpanzee social and mating behaviors as a basis for understanding early hominin social evolution. Discussing the absence of aggressive features in A. r. ramidus and its impact on understanding hominin social behavior, the researchers noted. Although A. r. ramidus is quite different from bonobos, with bonobos keeping a complex canine system, the shared reduction in sexual dimorphism and a more juvenile-like form compared to chimpanzees suggests that the social and developmental adaptations of bonobos may help in understanding early hominin social and sexual behavior. The trend towards more maternal care, female mate choice, and self-domestication might have been more pronounced and refined in A. r. ramidus than in bonobos. The researchers argued that many key human traits evolved in the late Miocene and early Pliocene forests and woodlands of Africa. Therefore, they proposed that humans may not have evolved from a chimpanzee-like ancestor as previously believed. This implies that many modern human traits represent deep evolutionary traits, and that chimpanzee behavior and physical features might have evolved after their divergence from the common ancestor shared with humans. Genus Australopithecus. The Australopithecus genus emerged in eastern Africa approximately 4 million years ago, gradually expanding across the continent until it went extinct around 2 million years later. During this era, several types of Australopiths were present, such as Australopithecus anamensis, O. Afarensis, O. Sediba, and O. Africanus. There is currently a dispute among scholars regarding whether certain African hominid groups from this era, like O. robustus and O. boisé, should be classified under the same genus. If they are, they would be categorized as O. robust Australopiths, whereas the remaining groups would be O. gracile Australopiths. On the other hand, if these groups are considered separate genera, they might be referred to as Paranthropus. Important genera and species from this time include Australopithecus, 4 to 1.8 million years ago O. Anamensis, O. Afarensis, O. Africanus, O. Barelghazali, O. Garhi, and O. Sediba. Kenyanthropus, 3 to 2.7 million years ago K. Platyops. Paranthropus, 3 to 1.2 million years ago P. Ethiopicus, P. boisé, and P. robustus. A newly proposed species, Australopithecus diaremida, is believed to have existed around the same time as O. afarensis. There is discussion on whether O. diaremida is a unique species or a variant of O. afarensis. Australopithecus prometheus, also known as Littlefoot, has been recently estimated to be 3.67 million years old using a novel dating method, placing this species on par with O. afarensis in terms of age. The presence of an opposable big toe in Littlefoot suggests it was an adept climber. Given the existence of nocturnal predators in its environment, it is hypothesized that Littlefoot constructed nesting platforms in trees at night, a behavior similar to that of chimpanzees and gorillas. Evolution of genus Homo. The first documented species of the genus Homo is Homo habilis, which appeared around 2.8 million years ago. It is recognized as the earliest known species with clear evidence of using stone tools. These early human ancestors had brains similar in size to chimpanzees, but it is theorized that this era saw the duplication of the human SRGAP2 gene, which likely led to faster development of the frontal cortex. For the next million years, there was a significant increase in brain size, and with the appearance of Homo erectus and Homo ergaster, the size of the cranium doubled to 850 cubic centimeters. Homo erectus and Homo ergaster are thought to be the first to master fire and create complex tools. And they were the pioneers in leaving Africa, spreading across Africa, Asia, and Europe between 1.3 and 1.8 million years ago. The latest theory on the origin of modern humans suggests that they evolved in Africa, possibly from H. heidelbergensis, H. rhodesiensis, or H. antecessor, and then migrated out of Africa between 50,000 and 100,000 years ago. They gradually replaced local populations of H. erectus, denisovans, H. floriensis, H. luzonensis, and H. neanderthalensis, whose ancestors had previously left Africa in earlier migrations. The ancestor of anatomically modern humans, known as archaic Homo sapiens, emerged during the Middle Paleolithic between 400,000 and 250,000 years ago. Recent genetic studies indicate that several haplotypes of Neanderthal origin are found in all non-African populations, suggesting limited interbreeding between these species. Neanderthals and other hominins, including denisovans, might have contributed up to 6% of their genome to modern humans. There are differing views among anthropologists regarding the shift to behavioral modernity, which is characterized by the emergence of symbolic culture, language, and specialized stone tool technology. Some believe this shift occurred around 50,000 years ago at the start of the Upper Paleolithic, while others propose a more gradual evolution over a longer period during the Middle Paleolithic. Homo sapiens is the only living species in the genus Homo. While some extinct Homo species may have been direct ancestors of Homo sapiens, many were likely cousins that diverged from the ancestral hominin lineage. Determining which groups should be classified as separate species or subspecies is challenging due to the limited number of fossils and the subtle differences used to classify species within the genus Homo. The Sahara pump theory, which explains the occasional presence of a wet Sahara desert, offers one explanation for the sporadic migration and speciation within the genus Homo. Research from the fields of archaeology and paleontology has shed light on the eating habits of different species of early humans and how their diet influenced their physical and behavioral development. Many experts in anthropology and archaeology back the Toba catastrophe theory, which argues that the massive eruption of Lake Toba in Indonesia around 70,000 years ago led to widespread famine, resulting in a significant reduction in the human population that impacted the genetic makeup of modern humans. However, the evidence for this theory is still controversial. A study published in 2023 indicates that a similar reduction in the human population might have happened between 930,000 and 813,000 years ago, lasting approximately 117,000 years, and nearly driving human ancestors to extinction. H. habilis and H. godingensis. Homo habilis existed from roughly 2.8 to 1.4 million years ago. This species evolved in regions of South and East Africa during the late Pliocene or early Pleistocene epoch, which spanned from about 2.5 to 2 million years ago. It branched off from the Australopithecines, developing smaller teeth and bigger brains. As one of the earliest recognized human ancestors, Homo habilis crafted tools from stones and possibly bones, which led to its nickname, Handy Man, by its discoverer, Louis Leakey. Some researchers suggest changing its classification to Australopithecus due to its body structure, which seems more adapted for life in trees rather than walking on two legs like later human ancestors. In May 2010, a new species, Homo godingensis, was identified in South Africa. H. ridulfensis and H. georgicus. Homo ridulfensis is the suggested name for a single, fragmented skull discovered in Kenya, with its connection to Homo habilis still uncertain. Homo georgicus, unearthed in Georgia, could signify a transitional species between Homo habilis and Homo erectus, or it might be a distinct subspecies of Homo erectus. H. ergaster and H. erectus. In 1891, Dutch doctor Eugene Du Bois uncovered the initial fossils of Homo erectus on the Indonesian island of Java. At first, Du Bois referred to these fossils as Anthropopithecus erectus, 1892 to 1893, thinking they resembled chimpanzees in appearance and behavior, and later as Pithecanthropus erectus, 1893 to 1894, believing their features were in between those of humans and apes. By the 20th century, German doctor and archaeologist Franz Weidenreich compared the features of Du Bois' Java man, then known as Pithecanthropus erectus, with those of Peking man, known as Sinanthropus pekinensis. In 1940, Weidenreich concluded that because of their anatomical similarities to modern humans, these fossils from Java and China should be classified under the same species of the genus Homo, specifically Homo erectus. Homo erectus existed from around 1.8 million years ago to approximately 70,000 years ago, possibly going extinct due to the Toba catastrophe, though the species Homo floresiensis managed to survive it. The initial stage of H. erectus, spanning from 1.8 to 1.25 million years ago, is sometimes seen as a distinct species, H. ergaster, or as Homo erectus ergaster, a subspecies of H. erectus. Nowadays, many experts use the term Homo ergaster for non-Asian forms of this group, while reserving H. erectus for those fossils from Asia that meet certain skeletal and dental criteria that are slightly different from H. ergaster. In Africa during the early Pleistocene, between 1.5 and 1 million years ago, certain groups of Homo habilis developed larger brains and began creating more complex stone tools. These distinctions were enough for anthropologists to classify them as a new species, Homo erectus, in Africa. The evolution of locking knees and the shift in the position of the foramen magnum likely played a role in these changes. This species might have also been the first to use fire for cooking meat. Richard Wrangham suggests that Homo was primarily terrestrial, with a shorter digestive system, smaller teeth, and larger, more energy-demanding brains. He proposes that the ability to control fire and cook food, which increased the nutritional value of food, was the main adaptation that set Homo apart from Australopithecines that lived in trees. H. sapronensis and H. antecessor. Various species have been suggested as transitional forms between H. erectus and H. heidelbergensis. H. antecessor remains found in Spain and England, range from 1.2 million to 500,000 years old. H. sapronensis. This term is used for a single skull found in Italy, believed to be around 800,000 years old. H. heidelbergensis. Heidelberg man occupied the Earth from about 800,000 to 300,000 years ago, also known as Homo sapiens heidelbergensis or Homo sapiens paleohungaricus. H. rhodesiensis and the Gauis cranium stand out as well. H. rhodesiensis. It's thought to have existed between 300,000 and 125,000 years ago. Currently, most experts place Rhodesian man within the Homo heidelbergensis category, though alternative classifications include archaic Homo sapiens and Homo sapiens rhodesiensis. In February 2006, the Gauis cranium was unearthed in Ethiopia, possibly indicating a species that lies between H. erectus and H. sapiens, or perhaps a separate evolutionary line. The skull, estimated to be between 500,000 and 250,000 years old, displays facial characteristics that hint at it being an intermediate species or a Bodo man, female. Further research is expected to be published in peer-reviewed journals. Neanderthal and Denisovan. Homo neanderthalensis, also known as Homo sapiens neanderthalensis, lived in Europe and Asia from around 400,000 to about 28,000 years ago. They had clear physical differences from anatomically modern humans, AMH, many of which were due to their better ability to survive in cold climates. Their ratio of surface area to volume was even lower than that of modern Inuit people, showing they were better at keeping warm. Neanderthals also had much larger brains, as shown by brain molds, challenging the idea that they were less intelligent than modern humans. However, their bigger bodies might have required larger brains for controlling movements. Recent research by Pierce, Stringer, and Dunbar has shown significant differences in brain structure. The bigger eye socket and back of the head in Neanderthals suggest they could see better than modern humans, which was an advantage in the darker conditions of Ice Age Europe. It's believed that Neanderthals used less brain space for social activities. Based on the size of their brain cavities, minus the back of the head, it's thought that Neanderthal groups could have had around 120 members, compared to about 144 for modern humans. Larger groups of modern humans might have meant less chance of inbreeding, wider trading networks, seen in the distribution of tools, and quicker sharing of social and technological advancements. These factors likely played a role in the eventual replacement of Neanderthal populations by modern Homo sapiens around 28,000 years ago. Initial studies of mitochondrial DNA suggested little gene exchange between H. neanderthalensis and H. sapiens, indicating they were separate species that split from a common ancestor around 660,000 years ago. However, in 2010, genome studies showed that Neanderthals did mate with anatomically modern humans about 45,000 to 80,000 years ago, right before modern humans moved out of Africa but before they spread across Europe, Asia, and beyond. Genetic analysis of a skeleton from Romania found that it contained 11% Neanderthal DNA, indicating an ancestor from 4 to 6 generations back, with earlier interbreeding in the Middle East. Although the Romanian group doesn't seem to be the direct ancestor of modern humans, it shows repeated instances of interbreeding. Today, non-African people carry 1% to 4%, or 1.5% to 2.6% based on recent studies, Neanderthal DNA, which matches with research showing the divergence of certain human genes about 1 million years ago, though this idea is still debated. Neanderthals and AMH Homo sapiens likely lived together in Europe for up to 10,000 years, during which time AMH populations grew quickly, potentially outnumbering and possibly outcompeting Neanderthals due to their larger numbers. In 2008, scientists digging in Denisova Cave in Siberia's Altai Mountains discovered a tiny bone piece from a young Denisovan, a unique human species. They also found artifacts, like a bracelet from the same layer, that were estimated to be around 40,000 years old. The cool conditions in the cave helped preserve the DNA in the fossil, enabling the sequencing of both mitochondrial DNA, mtDNA, and nuclear DNA. Unexpectedly, the mtDNA differences were ancient, yet full genome sequencing revealed that Denisovans were part of the same family as Neanderthals, splitting off from their common ancestor with modern humans shortly after. It's believed that modern humans shared time with Neanderthals in Europe and the Near East for possibly over 40,000 years, suggesting possible interactions and interbreeding among Neanderthals, Denisovans, and modern humans. This finding has complicated our understanding of human history during the late Pleistocene. Evidence indicates that Denisovans contributed up to 6% of the DNA in some modern Melanesians, showing limited interbreeding in Southeast Asia. Genetic research has pinpointed genetic markers from Neanderthals and Denisovans in the genomes of modern humans outside of Africa. HLA haplotypes from these ancient humans make up a large part of Eurasian HLA alleles, indicating a strong positive selection for these inherited markers. Corinne Simonidi at Vanderbilt University and her team have discovered that Neanderthal DNA segments might be linked to a higher incidence of depression in individuals of European descent, based on the medical records of 28,000 people. The flow of genes from Neanderthals to modern humans was not one way. In 2016, Sergi Castellano from the Max Planck Institute for Evolutionary Anthropology in Germany showed that Neanderthal genomes from Siberia are more similar to modern human genes than those from Europe, suggesting interbreeding around 100,000 years ago, likely in the Near East. Research on a Neanderthal child in Gibraltar suggests that Neanderthal children might have reached maturity faster than Homo sapiens, based on brain development and the timing of tooth eruption. H. floriensis. H. floriensis, which existed between 190,000 to 50,000 years ago, is often referred to as the Hobbit because of its small size, likely due to being isolated on an island. This species is intriguing not just for its small size but also for its relatively recent existence, belonging to the Homo genus with unique characteristics not seen in contemporary humans. While it shares a common ancestor with humans today, it evolved on a different evolutionary trajectory. The main finding, a skeleton thought to be that of a 30-year-old woman, was uncovered in 2003 and is estimated to be around 18,000 years old. This person was believed to be 1 meter tall, with a brain volume of only 380 cubic centimeters, smaller than that of a chimpanzee and less than a third of the average human brain size of 1,400 cubic centimeters. There is a current debate regarding whether H. floriensis is a separate species. Some researchers propose that it could be a modern human with a condition of pathological dwarfism, a theory somewhat supported by the existence of small populations on Flores, the Indonesian island where the skeleton was discovered. This, along with the condition of pathological dwarfism, could explain the significantly reduced size. Additionally, the tools found with H. floriensis are typically linked to H. sapiens. However, the theory of pathological dwarfism does not fully explain several anatomical differences between H. floriensis and modern humans, whether affected by disease or not, but that resemble those of ancient Homo species. These differences include unique cranial features and bone shapes in the wrist, forearm, shoulder, knee, and foot. Furthermore, the discovery of multiple individuals with these traits suggests these characteristics were widespread within a population rather than being isolated to one individual. In 2016, Australian archaeologist Gerrit van den Berg discovered fossil teeth and a partial jaw of hominins thought to be the ancestors of H. floriensis at Mata Menge, located about 74 kilometers (46 miles) from Liangbua. These fossils, dating back around 700,000 years, were noted by van den Berg for being even smaller than those found later. H. luzonensis. H. sapiens, which translates to wise or smart in Latin, first appeared in Africa around 300,000 years ago, likely coming from H. heidelbergensis or another related group. In September 2019, researchers employed computerized analysis of 260 CT scans to reconstruct the shape of the last shared ancestor of present-day humans, indicating that H. sapiens originated between 260,000 and 350,000 years ago from a combination of groups in East and South Africa. Between 400,000 and 250,000 years ago, during the Middle Pleistocene era, there was a rise in brain size and the emergence of sophisticated stone tool technologies, signifying the shift from H. erectus to H. sapiens. It's believed that H. erectus left Africa, and H. sapiens developed from H. erectus in Africa. After that, H. sapiens migrated both within and outside of Africa, eventually supplanting earlier H. erectus groups. This phenomenon is referred to as the recent single origin hypothesis or out of Africa theory. During these migrations, H. sapiens mated with ancient humans, such as Neanderthals and Denisovans in Eurasia. The theory suggesting a significant decrease in H. sapiens population around 70,000 years ago, known as the Toba catastrophe theory, was met with skepticism from the 1990s and gained minimal acceptance by the 2010s. The variety of human DNA is the result of the founder effect, mixing with ancient populations, and modern evolutionary forces. Anatomical changes. Since the species Homo sapiens split from its most recent common ancestor with chimpanzees, the process of human evolution has been characterized by a series of significant transformations in body structure, growth, physical functions, actions, and surroundings. The evolution of culture, which came about much later in the Pleistocene era, was pivotal in shaping human evolution, especially during shifts in ways of obtaining food. Key adaptations include walking on two legs, larger brains, longer periods of pregnancy and early childhood, and less pronounced differences between males and females. The connections among these changes are still subjects of current discussion. Other important physical changes include the development of a strong and precise grip, which was first seen in H. erectus. Bipedalism. Hominids developed bipedalism as their primary adaptation, leading to a series of skeletal modifications common among all bipedal hominids. The earliest hominids, possibly Sahelanthropus or Ororan, first appeared around 6 to 7 million years ago and exhibited basic bipedalism. At the same time, the knuckle walkers like gorillas and chimpanzees branched off from the hominin family tree, with Sahelanthropus or Ororan being our most recent common ancestor. Around 5.6 million years ago, Ardipithecus, a fully bipedal hominin, came into existence. Over time, early bipeds evolved into Australopithecines and eventually into the Homo genus. There are several theories regarding the benefits of bipedalism, including freeing the hands for tasks like gathering and carrying food, saving energy during movement, facilitating long-distance running and hunting, providing a wider field of vision, and reducing the risk of overheating by minimizing exposure to the sun. These adaptations were crucial for survival in the changing environments of the savannas and woodlands that emerged from the uplift of the East African Rift Valley. A 2007 study supports the idea that bipedalism evolved because it required less energy than knuckle walking. However, more recent research suggests that the ability to use fire was also necessary for the spread of humans across the globe. Bipedalism led to changes in limb proportions, with longer legs and shorter arms, reducing the need for brachiation. The big toe also aligned with the others, aiding in forward movement. Studies also show that Australopithecines continued to spend time in trees, as indicated by a grasping big toe, which gradually diminished in later hominins. The development of bipedalism brought about numerous skeletal modifications, affecting not only the legs and pelvis but also the spine, feet, ankles, and skull. The femur became more angled to shift the center of gravity towards the body center. The knee and ankle joints became stronger to support more weight. The spine became S-shaped, and the lumbar vertebrae became shorter and wider to support an upright posture. The foramen magnum shifted forward under the skull. The pelvic area underwent significant changes, with the iliac blade becoming shorter and wider to maintain balance while walking. This change resulted in a shorter but broader, bowl-like pelvis. However, this also led to a smaller birth canal compared to the knuckle-walking apes. Although the birth canal widened in modern humans compared to Australopithecines, allowing for the birth of larger-headed infants, further widening could have interfered with bipedal locomotion. The compact size of the birth canal and the shorter stature of the pelvis greatly influenced the process of human childbirth, making it more difficult than in other primates. The need for the fetal head to twist by about 90 degrees as it moves through the birth canal is a result of differences in pelvic dimensions. This smaller birth canal restricted the growth of the brain in early humans, resulting in shorter periods of pregnancy and the emergence of offspring that were less developed. Infants born to humans, who are not capable of walking until around 12 months, exhibit a higher degree of neoteny, or retention of juvenile characteristics, in comparison to other primates, who are able to move independently at a younger age. The expansion of the brain after birth and the increased reliance of children on their mothers had a profound effect on the female reproductive cycle, leading to a higher occurrence of alloparenting among humans as compared to other hominids. The phenomenon of delayed sexual maturation in humans also played a role in the development of menopause. The grandmother hypothesis proposes that older women were more likely to ensure the survival of their genetic lineage by looking after their grandchildren instead of reproducing more of their own offspring. Encephalization. The human species evolved to have significantly larger brains compared to other primates, with contemporary human brains averaging around 1,330 cubic centimeters, 81 cubic inches, which is nearly 3 times the volume of chimpanzee or gorilla brains. Following a period of little change with Australopithecus anamensis and Ardipithecus, which had smaller brains due to their ability to walk on 2 legs, the pattern of brain enlargement began with Homo habilis. Homo habilis had a brain volume of about 600 cubic centimeters, 37 cubic inches, slightly bigger than that of chimpanzees. This trend continued in Homo erectus, with brain volumes ranging from 800 to 1,100 cubic centimeters, 49 to 67 cubic inches, and reached its peak in Neanderthals, whose brains measured between 1,200 to 1,900 cubic centimeters, 73 to 116 cubic inches, larger than those of modern humans. This increase in brain volume was particularly noticeable after birth, with brain growth surpassing that of other apes, enabling longer periods of social learning and the development of language, starting around 2 million years ago. The growth in brain size may be connected to a dependence on food that is high in calories and difficult to obtain. The changes in the structure of human brains are arguably more important than the increase in size. Fossilized skulls show that early human brain sizes were within the range seen in modern humans 300,000 years ago, but the modern human brain shape only emerged between 100,000 and 35,000 years ago. The areas of the brain responsible for language, such as the temporal lobes, and those involved in complex decision-making and social behavior, like the prefrontal cortex, have grown significantly. Encephalization has been linked to a shift towards eating more starches and meat, although a 2022 study cast doubt on the significance of meat in this process. Other factors that may have contributed to these changes include the invention of cooking and the growing need to solve complex social issues as human societies became more intricate. Changes in the shape of the skull, such as smaller jaws and muscles, allowed for more space for brain growth. The expansion of the neocortex's volume, which included a rapid increase in the size of the cerebellum, traditionally known for its role in balance and fine motor skills but now also associated with speech and cognition, is particularly noteworthy. The great apes, including hominids, have a larger cerebellum in relation to the neocortex compared to other primates. The cerebellum's role in controlling sensory motor functions and learning complex muscle movements may have supported human technological advancements, including the development of speech. The direct survival benefits of encephalization remain uncertain, as the significant brain changes from Homo erectus to Homo heidelbergensis did not lead to major technological breakthroughs. These changes were likely social and behavioral, including enhanced empathy, larger social groups, and increased flexibility in behavior. Humans stand out for their ability to learn and adapt information through social learning. The emerging field of cultural evolution explores human socio-cultural changes from an evolutionary viewpoint. Ulnar opposition. Ulnar opposition, the skill of bringing the thumb to the little finger's tip on the same hand, is exclusive to the Homo genus, encompassing Neanderthals, the Cima de los Huesos hominins, and anatomically modern humans. In primates other than humans, the thumb is insufficiently long to reach the little finger. This ulnar opposition allows for the fine control and strength grips of the human hand, crucial for adept handling. Other changes. Human evolution has seen various transformations, such as a greater dependence on sight over smell. As shown by a significantly diminished olfactory bulb, a prolonged period of growth in youth and increased reliance on infants, a smaller digestive system and smaller, poorly aligned teeth, a quicker resting metabolic rate, a reduction in body hair, and a tenfold increase in the number of sweat glands in the skin compared to other primates of the catarrhine family. However, humans consume 30% to 50% less water daily than chimpanzees and gorillas. Moreover, humans undergo more rapid eye movement (REM) sleep but sleep less overall. The dental arch has shifted from a U-shape to a parabolic form, the emergence of a chin, a feature unique to humans, the existence of styloid processes, and a lower position of the larynx. As human hands and arms evolved for creating tools and lessened their ability to climb, the structure of the shoulder blades also changed, allowing our ancestors to throw objects with more power, speed, and precision. Use of tools. The employment of tools is frequently viewed as a sign of cleverness and may have been crucial in the development of human evolution, especially in the expansion of the human brain. Despite the brain's high demand for energy, it consumes approximately 13 watts, 260 kilocalories daily, or 1/5 of the body's energy at rest. Paleontology has yet to fully elucidate the mechanisms behind its growth over millions of years. The increased utilization of tools likely enabled early humans to pursue high-energy meat and process nutrient-rich plant foods, thereby creating a selective pressure for the enhancement of tool-making and usage abilities. Pinpointing the precise moment when early humans started using tools is difficult due to the nature of primitive tools, such as sharp-edged stones, which can be hard to differentiate from naturally occurring objects. There is some evidence to suggest that Australopithecines, around 4 million years ago, might have utilized broken bones as tools, although this theory is subject to debate. While many species create and use tools, humans have developed more sophisticated ones. The oldest known tools, flakes from West Turkana, Kenya, date back to 3.3 million years ago. The next oldest stone tools, from Gunna, Ethiopia, signify the start of Oldowan technology and are approximately 2.6 million years old. A Homo fossil discovered near Oldowan tools, dated at 2.3 million years old, implies that Homo species may have been the creators and users of these tools, though this claim is not yet conclusive evidence. The third metacarpal styloid process, which facilitates the locking of the hand bone into the wrist bone, provides humans with the dexterity and strength necessary for crafting and utilizing complex tools. This anatomical feature, which is absent in fossils older than 1.8 million years, sets humans apart from apes and other non-human primates. Bernard Wood noted that Paranthropus coexisted with early Homo species during the Oldowan industrial complex era. Although there is no direct proof linking Paranthropus to toolmaking, their skeletal structure suggests they might have had the capability to create tools. The majority of paleoanthropologists concur that early Homo species were primarily responsible for the creation of Oldowan tools, as these tools are often found in association with Homo fossils but not with Paranthropus. In 1994, Randall Sussman proposed that both Homo and Paranthropus species were involved in toolmaking based on the anatomy of their thumbs. He discovered that humans possess 3 thumb muscles absent in chimpanzees, along with thicker metacarpals with broader heads, which enhance precision in grasping. Sussman suggested that the modern human thumb structure evolved in response to the demands of toolmaking, indicating that both Homo and Paranthropus were indeed capable of toolmaking. Transition to behavioral modernity. Anthropologists describe contemporary human actions through a range of cultural and behavioral characteristics, including the development of specialized tools. The use of ornaments and images, like cave art, the arrangement of living areas, rituals, such as offering gifts in the afterlife, specific hunting methods, exploration of harsh environments, and trade networks based on exchanging goods. More broadly, these include the use of language and complex symbolic thought. There is a continuous debate regarding whether these actions were the result of a sudden revolution or a slow, gradual process. For the majority of the last 50,000 to 40,000 years, the advancement of stone tool technology was incremental, with each stage, H. habilis, H. ergaster, H. neanderthalensis, introducing new technologies that then developed slowly. Currently, there is discussion among paleoanthropologists about whether these Homo species displayed behaviors characteristic of modern humans, despite appearing to be culturally conservative, maintaining consistent technologies and foraging strategies over extended periods. Around 50,000 years ago, the evolution of human culture accelerated rapidly. This period, often referred to as the Great Leap Forward or the Upper Paleolithic Revolution, is characterized by the sudden emergence of behaviors indicative of modernity, such as hunting large game. However, evidence from Africa indicates that aspects of behavioral modernity appeared much earlier, with evidence of abstract art, varied ways of obtaining food, more advanced tools and weapons, and other modern behaviors. Many experts now believe that the shift to modern behavior occurred earlier than previously believed. Some researchers suggest a more gradual shift, pointing out that certain modern behaviors had already appeared among archaic Homo sapiens 300,000 to 200,000 years ago. Recent discoveries suggest that the Aboriginal population of Australia split from the African population 75,000 years ago and undertook a 160 kilometers, 99 miles, ocean voyage 60,000 years ago, which could question the importance of the Upper Paleolithic Revolution. Modern humans started to bury their dead, create clothing from animal hides, hunt with sophisticated methods, like using pit traps or driving animals off cliffs, and produce cave art. As human culture developed, different groups innovated upon existing technologies. Artifacts such as fish hooks, buttons, and bone needles show evidence of cultural diversity not seen before 50,000 BP. While older populations of H. neanderthalensis typically did not change their technologies, the Chattoperonian groups suggest Neanderthal imitations of H. sapiens or Ignatian technologies. Recent and ongoing human evolution. Human populations that are anatomically modern continue to evolve, influenced by both natural selection and genetic drift. While the selection pressure for certain traits, like resistance to smallpox, has decreased in recent times, humans still experience natural selection for many other characteristics. Some of these factors are specific to the environment, while others are due to changes in lifestyle since the advent of agriculture, 10,000 years ago, the growth of cities, 5,000 years ago, and the rise of industry, 250 years ago. It has been suggested that the pace of human evolution has quickened since the start of agriculture and civilization, leading to significant genetic differences among today's human populations. Recent studies have shown that human culture has introduced new forms of selection, which sometimes replace or work alongside natural selection. Examples of this include changes in superficial features like afro-textured hair, or the evolution of lighter skin and blonde hair in some groups, due to differences in climate. Humans have also adapted to high altitudes in various isolated groups, with Tibetans evolving over 3,000 years to have a high level of an EPAS1 allele that is advantageous at high altitudes. Evolutionary changes also include adaptations to endemic diseases. For instance, the presence of malaria favors the sickle cell trait, a heterozygous form of the sickle cell gene, whereas in the absence of malaria, the negative health effects of sickle cell anemia select against this trait. Another example involves the population at risk of Kuru, a severe disease, which shows a high prevalence of a specific immune variant of the prion protein gene G127V, likely due to the survival of immune individuals. Some evolutionary trends remain unexplained and are topics of current research in evolutionary medicine. For example, The prevalence of polycystic ovary syndrome, PCOS, which reduces fertility, suggests it should face strong negative selection, yet its high prevalence indicates a possible counteracting selection pressure, the nature of which is still being debated. Recent human evolution related to agriculture includes genetic resistance to infectious diseases from domesticated animals and metabolic changes from diet, such as the ability to digest lactose. Evolution driven by culture can sometimes contradict what natural selection would predict. For instance, while there is pressure for early reproduction, effective contraception, higher education, and changing social norms have led to selection in the opposite direction. However, culture-driven evolution can also align with natural selection. There are proposals that the recent expansion of the human brain may be due to a feedback loop where increased social learning efficiency leads to more complex cultural developments, which in turn drive further efficiency and brain expansion. Culture-driven evolution is also visible in the archaeological record. The development of stone tools during the Paleolithic period is linked to culturally driven cognitive development, with the acquisition of skills supported by culture and the evolution of increasingly complex technologies. In modern times, since the advent of industry, trends such as later onset of menopause have been observed. Other reported trends include longer reproductive periods and reductions in cholesterol levels, blood glucose, and blood pressure in some populations. History of study. Before Darwin. The word Homo, which is the scientific name for the human species, comes from the Latin word for human. Carl Linnaeus was the first to use this word in his system of classification. The Latin word humanus, which is the root of human, comes from the Indo-European root gem, which means earth. Linnaeus and his contemporaries also recognized the great apes as the nearest living relatives of humans, due to their shared physical features and structure. Darwin. The link between modern humans and our primate ancestors became clear following the release of Charles Darwin's seminal work, On the Origin of Species, in 1859. In this revolutionary piece, Darwin proposed that new species arise from existing ones through the process of evolution. Yet, his book did not provide detailed insights into the evolution of humans, merely suggesting that it would shed light on the origin of man and his history. The initial conversations on the topic of human evolution led to disagreements among Thomas Henry Huxley and Richard Owen. Huxley supported the idea that humans evolved from apes, pointing out both similarities and differences between humans and other primates, as he did in his 1863 publication Evidence as to Man's Place in Nature. At first, many of Darwin's early backers, including Alfred Russel Wallace and Charles Lyell, doubted that natural selection could fully account for the development of human intelligence and moral values. Yet, this viewpoint gradually changed. Darwin later incorporated his evolutionary theory, including the concept of sexual selection, into humans in his 1871 book, The Descent of Man, and Selection in Relation to Love. First fossils. During the 1800s, a major obstacle in understanding human evolution was the lack of fossil links in the field. In 1856, Neanderthal remains were discovered in a limestone mine, only 3 years before Charles Darwin's seminal work, On the Origin of Species, was published. Earlier finds in Gibraltar were mistakenly thought to be of modern humans suffering from disease. The discovery of Homo erectus by Eugene Du Bois in 1891 at Trinil, Java, was a significant breakthrough, but it wasn't until the 1920s, with the discovery of more fossils in Africa, that evidence of intermediate species started to emerge. The 1925 identification of Australopithecus africanus by Raymond Dart, from the Tong child, was a critical point. This young Australopithecine was found in a cave, with a remarkably preserved small brain, 410 cubic centimeters, with an endocast similar to that of humans, not chimpanzees or gorillas. The individual had short canine teeth and a skull position that suggested it walked on 2 legs. Dart believed the Tong child was a direct ancestor of bipedal humans, linking the evolutionary chain from apes to humans. The East African fossils. In the 1960s and 1970s, the Leakey family, including Louis and Mary Leakey, their son Richard, and daughter-in-law Maeve, made significant contributions to the field of paleontology through their extensive fossil discoveries in East Africa, especially at the Olduvai Gorge and Lake Turkana. They carefully dug up fossil sites in these areas, revealing a variety of early human species such as Australopithecines, different Homo species, and Homo erectus. These discoveries firmly established Africa as the cradle of humanity. By the late 1970s and early 1980s, Ethiopia became a key location in the study of human evolution with the discovery of Lucy, in 1974 by Donald Johanson near Hadar in the dry Afar Triangle of northern Ethiopia. Despite having a relatively small brain, Lucy's pelvic and leg bones were nearly identical to those of humans, proving that bipedal walking was a characteristic of these early hominins. Lucy was identified as Australopithecus afarensis, a species believed to be closely related to Homo, possibly a direct ancestor or a close relative of an ancestor from that early era. The name Lucy was chosen in honor of the Beatles' song Lucy in the Sky with Diamonds, which was played often and loudly during the excavation site. Further excavations in the Afar Triangle, led by Tim D. White in the 1990s, uncovered additional hominin fossils, including those of Ardipithecus ramidus and Ifkadaba. In 2013, the discovery of fossil skeletons belonging to Homo naledi in the Rising Star cave system near Johannesburg in South Africa's Cradle of Humankind region shed light on an extinct hominin species. These hominins, thought to be part of the Homo genus, had a body size and height similar to that of small modern humans, a brain size similar to Australopithecus, and cranial features similar to early Homo species. Their skeletal structure showed a mix of primitive traits from Australopithecines and early hominins. Notably, these individuals were found to have been buried in the cave, dating back to around 250,000 years ago, which places them at the same time as the emergence of larger-brained anatomically modern humans. The genetic revolution. The study of human evolution took a significant leap forward with the groundbreaking work of Vincent Sarek and Alan Wilson. They pioneered the use of immunological cross-reactions in blood serum albumin to measure the relatedness among different species, including humans and African apes like chimpanzees and gorillas. By quantifying these reactions as immunological distances, which were linked to the number of amino acid differences in similar proteins across species, they laid the foundation for a molecular clock. This clock was designed to estimate the time when species diverged, especially for those with limited or no fossil records. In their seminal 1967 article in Science, Sarek and Wilson estimated that humans and apes had diverged between 4 to 5 million years ago. This estimate was in contrast to previous interpretations from the fossil record, which suggested a much older divergence, possibly as long as 30 million years. However, the discovery of fossils like Lucy and a re-examination of older fossils such as Ramapithecus lent support to their younger estimates and confirmed the reliability of the albumin method. The field of human origins was further advanced by the sequencing of DNA, particularly mitochondrial DNA, mtDNA, and Y-chromosome DNA, yDNA. The application of the molecular clock concept transformed the study of molecular evolution. Early studies based on the molecular clock, which indicated that orangutans had split around 10 to 20 million years ago, suggested a mutation rate of about 76 mutations per generation that were not inherited by offspring. This supported the earlier divergence estimates between hominins and chimpanzees. However, a 2012 study in Iceland, which involved 78 parent-child pairs, suggested a lower mutation rate of around 36 mutations per generation. This new finding pushed back the divergence between humans and chimpanzees to an earlier time, possibly over 7 million years ago. Further research, including the analysis of genes from wild chimpanzee populations, indicated that chimpanzees reach reproductive maturity at around 26.5 years old. This information suggested that the split between humans and chimpanzees occurred between 7 and 13 million years ago. It also proposed that species such as Ardipithecus, 4.5 ma, or Orin, 6 ma, and Sahelanthropus, 7 ma, might be part of the hominid lineage, possibly outside the East African Rift area where earlier theories had placed the split. Additionally, genetic studies in 2006 revealed that following the initial divergence of the human and chimpanzee lineages, there was evidence of interspecies mating between early humans and chimpanzees. This interbreeding resulted in genetic changes in the gene pools of both species, with the X chromosomes of humans and chimpanzees diverging about 1.2 million years more recently than other chromosomes. This discovery challenged conventional views and underscored the complex evolutionary history shared between humans and their closest living relatives. The quest for the earliest hominin. In the 1990s, numerous groups of paleoanthropologists carried out thorough investigations across Africa to find proof of the first split of the hominin lineage from the great apes. In 1994, Meev Leakey found a crucial piece of evidence with Australopithecus annamensis. Yet, this achievement was somewhat dimmed by Tim D. White's 1995 find of Ardipithecus ramidus, which moved the timeline of the fossil record back to 4.2 million years ago. By 2000, Martin Pickford and Bridget Sennett discovered a human-like creature that walked on two legs in Kenya's Tugen Hills, which they dated to 6 million years ago and called it Ororantogenensis. Later that year, Michel Brunet's group found the skull of Sahelanthropus cadensis, estimated to be 7.2 million years old. Brunet believed Sahelanthropus walked on two legs, placing it in the category of hominids and more specifically, hominins, referencing hominidae. Human dispersal. During the 1980s, experts in anthropology had various opinions about the barriers to reproduction and the movement patterns of the Homo genus. These discussions were later clarified by genetic research, offering new understandings of human development. The Sahara pump theory suggests that Homo species left Africa at least 3, and possibly 4, times, including Homo erectus, Homo heidelbergensis, and several groups of Homo sapiens. Recent discoveries show a strong connection between these movements and times of changing climate. New evidence is now pointing to the possibility that humans may have left Africa earlier than thought. A joint effort between French and Indian researchers found human remains in the Siwak Hills, north of New Delhi, that are at least 2.6 million years old. This discovery is older than the earliest Homo fossils found in Manasai, Georgia, which are dated to 1.85 million years ago. Debated discoveries from a Chinese cave also support the idea of early tool use, with tools potentially as old as 2.48 million years. This indicates that the Asian chopper tool tradition, seen in Java and northern China, might have evolved before the Acheulean hand axe was made in Africa. Dispersal of modern Homo sapiens. Prior to the discovery of genetic proof, there were two primary theories regarding how modern humans spread across the globe. The multi-regional hypothesis, first introduced in 1988 by Milford H. Woolpoff, suggested that the Homo genus was made up of a single, unified group, rather than separate species, that evolved globally over the last several million years. On the other hand, the out of Africa theory, developed by Chris Stringer and Peter Andrews, suggested that modern H. sapiens emerged relatively recently in Africa, around 200,000 years ago, and then spread across Eurasia, eventually almost completely replacing other Homo species. Analyzing mitochondrial DNA, mtDNA, and Y-chromosomal DNA from various indigenous groups has shed light on our ancestral history, strengthening the out of Africa hypothesis and reducing the evidence for a widespread evolution from different regions. The genetic differences observed in evolutionary trees are seen as proof for a recent, single origin. The out of Africa hypothesis has been significantly backed by research on mtDNA and the male Y chromosome. By examining genealogical trees based on 133 mtDNA types, it has been suggested that all humans share a common African female ancestor, known as mitochondrial Eve. Moreover, the diversity of mtDNA is highest in African populations, further supporting the idea of an African origin. A detailed study by Sarah Tishkoff on the genetic diversity of Africa found the San people to have the highest genetic diversity among 113 different populations, placing them within 14 ancestral groups. This research indicates a possible origin of modern humans near the coast of Namibia and Angola in southwestern Africa. Although fossil evidence has not conclusively determined the first appearance of modern humans in Africa, research on haplogroups in Y-chromosomal DNA and mtDNA consistently points to a recent African origin. Autosomal DNA studies also largely support this view. However, the sequencing of Neanderthal and Denisovan genomes has shown some interbreeding with these ancient hominins, with modern human populations outside Africa inheriting 1 to 4% of Neanderthal DNA and Melanesians carrying 4 to 6% of Denisovan DNA. These discoveries, while challenging the most stringent interpretations of the out of Africa theory, suggest that following a genetic bottleneck, possibly related to the Toba supervolcano eruption, a small group of humans left Africa, intermingled with Neanderthals in areas like the Middle East or Eurasian steppe, and then with Denisovans in Southeast Asia. The descendants of these interactions spread across the globe, with modern Eurasian and Oceanian populations showing genetic traces of both Neanderthals and Denisovans. There's an ongoing discussion about whether early humans left Africa in one big exodus or in several smaller waves. The idea of multiple dispersal includes the southern dispersal theory, which has become more accepted due to genetic, linguistic, and archaeological evidence over the past few years. This theory suggests that about 70,000 years ago, modern humans spread out along the coast from the Horn of Africa, moving across the Bab el-Mandeb to Yemen when the sea levels were lower. This group then went on to settle in Southeast Asia and Oceania, which explains why there are early human sites in these areas that are older than those in the Levant. Their way of life was very much centered around the sea. Stephen Oppenheimer has suggested a later wave of human migration, possibly through the Persian Gulf oases and the Zagros Mountains into the Middle East, or maybe by crossing the Sinai Peninsula into Asia around 50,000 years ago. This later group might have had more sophisticated technology for hunting large animals and might have relied less on sea resources than the first group. The evidence for the first group's spread might have been hidden by the sea levels rising at the end of the last ice ages. However, studies on mitochondrial DNA L3 lineages show that all modern non-African populations, including those in Eurasia and Oceania, share a common ancestor from Africa between 65,000 and 50,000 years ago. This genetic data challenges the idea of multiple dispersal, suggesting instead that all non-African human populations have a single origin. Evidence. The comprehension of human evolution is built upon a variety of natural science disciplines. Traditionally, the examination of fossils has been the main resource, offering valuable clues about the mechanisms of evolution. Yet, with the introduction of genetics in the 1970s, the analysis of DNA has emerged as equally important. Research in the development of organisms, their evolutionary relationships, and the study of evolutionary developmental biology across both vertebrates and invertebrates has also been crucial in enhancing our knowledge of evolution, including that of humans. Anthropology, especially paleoanthropology, is essential in investigating the prehistoric life of humans and the beginnings of human existence. Evidence from genetics. Humans are closely related to bonobos and chimpanzees, both in the Pan genus, as well as gorillas, in the gorilla genus. The comparison of human and chimpanzee DNA has shown a high degree of similarity, ranging from 95% to 99% by 2012. By using the molecular clock method, which estimates the time it takes for genetic differences to accumulate between species, researchers can approximate when the human and chimpanzee ancestors split. Gibbons, from the Hylobatidae family, and orangutans, from the Pongo genus, were the first to branch off from the lineage that eventually led to hominins, including humans, followed by gorillas, from the gorilla genus, and then chimpanzees, from the Pan genus. The split between hominins and chimpanzees is thought to have occurred between 4 to 8 million years ago during the late Miocene. The process of speciation took a long time, with the initial divergence happening between 7 to 13 million years ago. Hybridization prolonged the separation over several million years, with Patterson, 2006, suggesting the final split around 5 to 6 million years ago. Research within the Homo genus has looked into the exchange of genes between early modern humans and Neanderthals, which has deepened our understanding of early human migration routes and the dates of divergence. By examining parts of the genome that evolved non-selectively, it's possible to construct a genetic tree that covers the entire human species since their last common ancestor. Every time a new mutation, single nucleotide polymorphism, appears in an individual and is passed down to their offspring, a haplogroup is formed, including all individuals who carry that mutation. The study of mitochondrial DNA, which is passed down exclusively from the mother, can trace back to mitochondrial Eve, the most recent common maternal ancestor of all modern humans, who is believed to have lived around 200,000 years ago. The field of human evolutionary genetics explores the differences in DNA among individuals, their evolutionary origins, and their significance in anthropology, medicine, and forensics. Genetic data continues to offer vital insights into human evolution. Recent research from May 2023 indicates a more complex human evolutionary history than previously believed, suggesting diverse origins across various times and places within Africa. Evidence from the fossil record. There's a scarcity of fossil proof concerning when the gorilla, chimpanzee, and human-like species began to diverge. Suggested early human-like species include Sahelanthropus cadensis from 7 million years ago, or Orantugenensis from 5.7 million years ago, and Ardipithecus cadaba from 5.6 million years ago. Each has been proposed as a human-like ancestor, though these theories are contested. It's also possible that these species might belong to a different group of African apes or represent a shared ancestor with humans. The precise connection between these early human-like species and the human lineage is still unclear. Around 4 million years ago, the Australopithecines appeared, splitting into two main groups, robust, Paranthropus, and gracile. One group, possibly including A. garhi, is thought to have given rise to the ancestors of the Homo genus. Within the Australopithecines, Australopithecus afarensis is well documented with over 100 individuals discovered from northern Ethiopia, like the well-known Lucy, to Kenya and South Africa. Fossils of larger Australopithecines like O. robustus, or Paranthropus robustus, and O/P. boisé are plentiful in South Africa, for example, at Kromdrai, Swartkrans, and around Lake Turkana in Kenya. The first species of the Homo genus, Homo habilis, appeared around 2.8 million years ago. Homo habilis is the first to show clear signs of using stone tools, which led to the development of the Oldowan lithic technology named after the Oldoway Gorge. Some experts believe Homo rudolfensis, a group with a larger body size like early H. habilis, is a distinct species, while others argue they are part of H. habilis, indicating intraspecies variation or sexual dimorphism. These early hominins had brain sizes similar to chimpanzees and mainly adapted through bipedalism for life on land. Over the next million years, the process of encephalization began, and by the emergence of H. erectus around 1.9 million years ago, their cranial capacity had doubled. H. erectus was the first hominin to leave Africa, spreading across Africa, Asia, and Europe from 1.8 to 1.3 million years ago. A group of H. erectus, sometimes referred to as H. ergaster, stayed in Africa and evolved into H. sapiens. H. erectus and H. ergaster are believed to be the first to use fire and create complex tools. The genetic revolution. The genetic breakthrough in understanding human evolution kicked off with Vincent Serrick and Alan Wilson, who led the way in measuring the immune system reactions of blood serum albumin across various species, including humans, chimpanzees, and gorillas. These reactions introduced a quantitative measure called immunological distance, ID, which was linked to the number of amino acid differences in similar proteins across species. By creating a calibration curve for ID against known divergence times from fossils, Serrick and Wilson crafted a molecular clock to estimate the times of divergence for species lacking complete fossil records. In their groundbreaking 1967 article in Science, Serrick and Wilson put forward an estimate of 4 to 5 million years for the split between humans and apes, challenging the earlier views from the fossil record that suggested a much older separation, ranging from 10 to 30 million years. Subsequent fossil finds, like Lucy, and reinterpretations of older fossils, such as Ramapithecus, backed up these newer estimates and confirmed the reliability of the albumin method. The development of DNA sequencing, especially for mitochondrial DNA, mtDNA, and later Y chromosome DNA, y-DNA, greatly deepened our understanding of human origins. The use of the molecular clock concept transformed the field of molecular evolution. Early molecular clock studies, which placed the split from orangutans around 10 to 20 million years ago, initially suggested a high mutation rate that supported the divergence time between hominins and chimpanzees mentioned earlier. However, a 2012 study in Iceland involving 78 parent-child pairs proposed a slower mutation rate of about 36 mutations per generation, pushing the divergence between humans and chimpanzees to over 7 million years ago, ma. Additional research with wild chimpanzee populations indicated an average age at reproduction of about 26.5 years, further reinforcing a divergence period between 7 and 13 million years ago. This timeline suggests that Ardipithecus, 4.5 ma, Ororan, 6 ma, and Sahelanthropus, 7 ma, might all be part of the hominin lineage, possibly outside the East African Rift area. Furthermore, a 2006 study on genes from both species showed evidence of interspecies mating after the initial split. Recent genomic comparisons between humans and chimpanzees hint at ongoing interbreeding. Notably, the X chromosomes of humans and chimpanzees diverged about 1.2 million years later than other chromosomes. This research suggests there were two distinct splits in the human-chimpanzee lineage, with initial interbreeding followed by a second split. These discoveries have captivated paleoanthropologists, leading to a closer examination of the new genetic data. The quest for the earliest hominin. In the 1990s, numerous groups of paleoanthropologists carried out thorough investigations across Africa looking for clues that indicate the earliest split of the human ancestor group from the great apes. In 1994, Meave Leakey found a crucial piece of evidence with Australopithecus annamensis. Yet, this breakthrough was somewhat eclipsed by Tim D. White's 1995 find of Ardipithecus ramidus, which pushed back the timeline of the fossil record to 4.2 million years ago. By 2000, Martin Pickford and Bridget Sennett discovered a 6-million-year-old human ancestor in Kenya's Tugen Hills, which they named Ororan tugenensis. Later that year, Michel Brunet's group revealed the skull of Sahelanthropus cadensis, estimated to be 7.2 million years old. Brunet suggested that this find was proof of walking on two legs, classifying Sahelanthropus cadensis as a member of the human ancestor family, specifically a hominin, hominidae, terms hominids and hominins. Human dispersal. During the 1980s, experts in anthropology had a variety of opinions regarding the barriers to reproduction and the movements of species within the Homo genus. However, the field of genetics has been instrumental in settling these disagreements. The evidence in favor of the Sahara pump theory suggests that Homo species left Africa on several occasions, possibly as many as 3 or 4, including Homo erectus, Homo heidelbergensis, and Homo sapiens. These migrations seem to be connected to times of changing climate. New findings suggest that humans might have left Africa earlier than was previously thought. A collaboration between French and Indian teams discovered human remains in the Siwak Hills, north of New Delhi, that are at least 2.6 million years old, which is older than the earliest Homo discovery at Manasai, Georgia, which was 1.85 million years old. The controversial discovery of tools in a Chinese cave also supports the idea that humans began using tools as early as 2.48 million years ago. This implies that the Asian chopper tool tradition, found in Java and northern China, could have started before the Acheulean hand axe was invented in Africa. Dispersal of modern Homo sapiens. Prior to the emergence of genetic proof, there were two main theories regarding how modern humans spread across the globe. The first, known as the multi-regional hypothesis, was proposed by Milford H. Woolpoff in 1988. This theory suggested that Homo sapiens originated from a single, interconnected group that evolved globally over millions of years without clear divisions into different species. On the other hand, the out of Africa theory, developed by Chris Stringer and Peter Andrews, argued that Homo sapiens evolved into distinct species in Africa around 200,000 years ago, and then spread throughout Eurasia, largely replacing other hominin species. The discovery of mtDNA and Y-DNA from various native populations played a crucial role in reinforcing the out-of-Africa theory and questioning the concept of multi-regional evolution. Genetic studies, including the creation of genealogy trees using 133 different mtDNA types, traced all maternal lineages back to a common African ancestor, known as mitochondrial Eve. This theory is further backed by the highest levels of mitochondrial genetic diversity seen in African populations. Sarah Tishkoff's in-depth research on the genetic diversity of Africa highlighted the San people as having the highest genetic diversity among 113 different populations studied. This research indicated that modern human migration likely began in southwestern Africa, near the coast of Namibia and Angola. Although there is not enough fossil evidence to definitively pinpoint the location of the first appearance of modern humans in Africa, genetic studies of Y-chromosomal DNA and mtDNA consistently point to an African origin for the modern human lineage. Although evidence from autosomal DNA mostly supports an African origin for modern humans, recent research has suggested that there was interbreeding between modern humans and ancient groups such as Neanderthals and Denisovans. The genetic analysis of Neanderthal and Denisovan DNA shows that all non-African human groups carry genetic markers from Neanderthals, with additional Denisovan genetic markers being more common among Melanesians. These findings complicate the out-of-Africa theory, indicating that there may have been interbreeding events following the initial migration out of Africa, possibly due to genetic bottlenecks and environmental conditions like the Toba supervolcano eruption. In conclusion, while the out-of-Africa theory is generally accepted by genetic evidence, the complexities of human migration and interbreeding highlight the ongoing process of refining our understanding of ancient human populations. There is ongoing discussion about whether the movement of humans out of Africa happened in one big exodus or through several smaller dispersals. A well-known theory, the Southern Dispersal Model, has gained backing from genetic, linguistic, and archaeological studies in recent times. This theory suggests that approximately 70,000 years ago, modern humans spread along the coast from the Horn of Africa, moving across the Bab el-Mandeb, a strait between the Red Sea and the Gulf of Aden, during a time when sea levels were lower. This group then went on to populate Southeast Asia and Oceania, explaining the early discovery of human sites in these areas compared to the Levant. It is believed that they heavily depended on sea resources for survival. Stephen Oppenheimer proposed a later migration through areas like the Persian Gulf oases and the Zagros Mountains into the Middle East. Alternatively, this group might have crossed the Sinai Peninsula into Asia around 50,000 years ago, playing a significant role in the spread of human populations across Eurasia. It's suggested that this later group might have had more advanced tools for hunting large animals and was less dependent on coastal food sources than the earlier group. Evidence for the first group's expansion might have been hidden by rising sea levels at the end of glacial periods. However, research on mitochondrial DNA L3 lineages shows that all modern non-African populations, including those in Eurasia and Oceania, share a common ancestor from a single migration out of Africa between 65,000 and 50,000 years ago. This genetic evidence challenges the idea of multiple dispersals, indicating a single origin for all non-African populations. In conclusion, while the debate continues, genetic evidence strongly points to a single major migration event from Africa as the main source of modern human populations outside of Africa. Interspecies breeding. The concept of interbreeding, also referred to as hybridization or mixing, has been a topic of discussion since the 19th century, following the discovery of Neanderthal remains. The traditional view of human evolution, which suggested a straightforward, linear path, began to lose its appeal in the 1970s with the identification of various human species, making the notion of a direct, linear evolution increasingly doubtful. In the 21st century, breakthroughs in molecular biology and computational methods have allowed for the complete sequencing of the genomes of Neanderthals and humans, providing evidence of recent interbreeding among different human groups. By 2010, evidence from molecular biology confirmed instances of interbreeding during the Middle Paleolithic and early Upper Paleolithic eras. It was demonstrated that these interbreeding events occurred independently among Neanderthals, Denisovans, and possibly other unknown human ancestors. Currently, about 2% of the DNA in non-African populations, including Europeans, Asians, and Oceanians, comes from Neanderthals, with additional traces of Denisovan DNA found in modern Melanesians, ranging from 4% to 6%. Comparing the human genome to that of Neanderthals, Denisovans, and other apes helps identify unique characteristics that set modern humans apart from other hominin species. For example, research in the mid-2010s uncovered a variety of traits related to neurological, immunological, developmental, and metabolic functions that were developed by archaic humans in European and Asian settings and were inherited by modern humans through interbreeding with local hominin populations. Despite ongoing debates about the story of human evolution, findings since 2010 have highlighted that human evolution is not a straightforward, linear, or branching process, but rather a complex interaction among related species. Genomic studies suggest that interbreeding between lineages that were significantly diverged played a key role in the emergence of modern humans. Stone tools. The earliest known stone tools were discovered around 2.6 million years ago, when early humans in Eastern Africa began making basic tools called choppers from flat stones by striking them with other stones. This event is considered the start of the Paleolithic, or Old Stone Age, which lasted until about 10,000 years ago, marking the end of the last ice age. The Paleolithic period is further broken down into three stages: the Lower Paleolithic, Early Stone Age, which ended around 350,000 to 300,000 years ago, the Middle Paleolithic, Middle Stone Age, which ended between 50,000 and 30,000 years ago, and the Upper Paleolithic, Late Stone Age, which lasted from 50,000 to 10,000 years ago. Researchers digging in the Great Rift Valley of Kenya have found the oldest known stone tools in the world, dating back to around 3.3 million years. These tools are even older than the oldest known stone tools from Ethiopia, which were dated to be around 700,000 years old. Between 700,000 and 300,000 years ago, the Acheulean period is known for the creation of large stone hand axes by early humans, such as H. ergaster, or erectus, using materials like flint and quartzite. At first, these axes were crude, but they became more refined over time through a process called retouching, which involved making additional strikes along the edges of the flakes. Around 350,000 years ago, a more advanced technique called the Levallois method was developed, which included precise strikes to make tools like scrapers, slicers, roqueloirs, needles, and flattened needles. By about 50,000 years ago, Neanderthals and Cro-Magnons, early modern humans, were making increasingly sophisticated and specialized stone tools like knives, blades, and skimmers. There is also evidence that early humans in Africa were making bone tools as early as 90,000 to 70,000 years ago, and these tools have been found at early human sites in Eurasia dating back to about 50,000 years ago. Great job, everyone, for taking the time to watch this video. We're sure it was both educational and captivating. Please remember to leave your comments before the clock runs out. Your input is incredibly valuable to us. If you're just joining us, don't forget to subscribe to our channel by clicking the bell icon to ensure you never miss any of our updates. And if you liked what you saw, please let us know by giving it a thumbs up. It truly aids us in reaching a wider audience like you. Spread the word among your friends and family to help spread awareness and initiate important discussions. Together, we can create a change. Thanks again for your attention. Be on the lookout for more exciting content right here soon.